We live in an age of unprecedented scientific advancement. Every day, new discoveries push the boundaries of what we thought possible. Biotechnology, fueled by groundbreaking discoveries in genetics and genomics, offers remarkable potential to improve human health, agriculture, and the environment. From curing genetic diseases to enhancing crop yields, the possibilities seem endless. But this immense power to reshape life itself raises profound ethical questions. How far should we go in altering the very fabric of life? Bioethics provides the essential framework for navigating these complex issues. It helps us weigh the moral implications of our scientific endeavors. It compels us to grapple with the potential consequences of our actions, ensuring that we consider not just the immediate benefits, but also the long-term impacts. And it ensures that scientific progress aligns with human values, promoting a balance between innovation and ethical responsibility. Bioethics is not about halting progress. It's about guiding it in a direction that is beneficial and just. It's about steering it responsibly, ensuring that our advancements serve the greater good. When we consider the implications of gene editing, we must think about the potential for both positive and negative outcomes. The use of genetically modified organisms in agriculture can lead to more resilient crops, but it also raises concerns about biodiversity and ecosystem balance, or the storage and use of our genetic data, which brings up issues of privacy and consent. Who controls this data and how is it used? We must ask ourselves, what are the risks and benefits? How do we ensure that the benefits outweigh the risks? Who stands to gain or lose from these advancements? Are we considering the needs and rights of all communities? What are our ethical obligations to future generations? How do we ensure that our actions today do not harm those who come after us? These are not easy questions. They require deep reflection and a willingness to engage in difficult conversations. They demand thoughtful consideration from scientists, ethicists, policymakers, and the public alike. Only through collaboration can we find balanced solutions. By engaging in open and informed dialogue, we can harness the power of biotechnology while safeguarding our values and ensuring a future where scientific progress benefits all humankind. We can celebrate the breakthroughs and innovations that biotechnology brings, knowing that we have considered their ethical implications. It's the dawn of a new era, an era filled with promise and potential, an era where we can rewrite the code of life, opening up possibilities we never dreamed of. But with great power comes great responsibility. We must tread carefully and thoughtfully. Let's proceed with wisdom and caution, ensuring that our steps forward are guided by ethical considerations and a commitment to the greater good. Gene editing technologies, particularly CRISPR-Cas9, have revolutionized genetic engineering. This powerful tool allows scientists to cut and modify DNA with unprecedented precision. We now have the ability to make precise changes to DNA sequences, offering the potential to cure genetic diseases, enhance crop yields, and even reshape entire ecosystems. The implications of this technology are vast and far-reaching. This groundbreaking technology holds immense promise for addressing some of humanity's most pressing challenges. From agriculture to medicine, the applications are seemingly endless. Imagine a world without genetic diseases, cystic fibrosis, Huntington's disease, sickle cell anemia. These debilitating and often fatal conditions could become relics of the past. The potential to eradicate such diseases is within our grasp. Gene editing offers a glimmer of hope for millions affected by these diseases. It could lead to treatments that are more effective and less invasive than current options. It promises to alleviate suffering and transform lives. Families could be freed from the burden of hereditary illnesses, and individuals could lead healthier, longer lives. But this potential comes with significant risks. The power to alter DNA is not without its dangers. The long-term effects of gene editing are still unknown. We are venturing into uncharted territory, and the consequences could be profound. Unintended mutations could have unforeseen consequences, potentially creating new diseases or disrupting delicate ecosystems. The balance of nature could be altered in ways we cannot predict. The ethical implications of altering the human germline, the DNA passed down to future generations, are profound and demand careful consideration. The decisions we make today will affect countless future generations. As we stand on the cusp of this genetic revolution, we must proceed with a balance of optimism and caution. The promise of gene editing is matched by its peril. The potential benefits of gene editing are undeniable, but so too are the risks. We must weigh the possibilities against the potential for harm. Responsible innovation requires careful assessment, ethical oversight, and ongoing public dialogue. Only through a collaborative and thoughtful approach can we harness the power of gene editing for the greater good while minimizing its risks. The first principle of medicine, do no harm, applies equally to the realm of gene editing. Before we can unleash the full potential of this technology, we must ensure its safety and efficacy. Rigorous research and clinical trials are essential to identify and mitigate potential risks. Unintended consequences are a major concern with gene editing. The human genome is incredibly complex. Altering one gene could have unforeseen effects on other genes or biological pathways. These off-target edits could lead to new diseases or exacerbate existing ones. Moreover, the long-term effects of gene editing are largely unknown. Will edits made to somatic cells, the non-reproductive cells of the body, have lasting consequences? Could germline editing, which alters the DNA passed down to future generations, have unintended and irreversible impacts on the human gene pool? These are not merely theoretical concerns. We must proceed with caution, prioritizing patient safety and well-being above all else. Thorough preclinical testing, careful monitoring of clinical trials, and long-term follow-up are crucial to ensuring the responsible development and deployment of gene editing technologies. Section 4. Access and equity, ensuring fairness in genetic advancements. The potential benefits of gene editing should be accessible to all, not just the privileged few. Ensuring equitable access to these transformative technologies is a moral imperative. We must strive to prevent a future where the genetically enhanced have an unfair advantage over the genetically unenhanced. The cost of developing and delivering gene therapies is currently prohibitive for many. If left unchecked, this could create a two-tiered healthcare system where the wealthy can afford to correct genetic flaws while the less fortunate are left behind. Furthermore, access to gene editing technologies should not be limited by geographical location or socioeconomic status. We need to ensure that these advancements benefit people in both developed and developing countries, bridging the gap in healthcare disparities. Promoting global collaboration, fostering technology transfer, and implementing equitable pricing models are crucial steps towards ensuring that the fruits of gene editing are shared by all. We must strive for a future where these transformative technologies are a tool for social justice, not a driver of inequality. Section 5. Consent and germline editing, shaping future generations. Gene editing raises profound ethical questions about consent, particularly when it comes to germline editing. This type of editing involves making changes to the DNA of embryos, which means these changes will be passed down to future generations. The implications of such actions are vast and complex. Do we have the right to alter the genetic makeup of future generations? This question is at the heart of the ethical debate surrounding germline editing. The potential to eradicate genetic diseases is promising, but it also opens the door to a host of moral dilemmas. Who gets to decide which traits are desirable and which are not? This decision-making process is fraught with ethical concerns. The power to alter human genetics could be misused, leading to a society where certain traits are valued over others, potentially reinforcing harmful biases. Informed consent is a cornerstone of medical ethics. It ensures that patients are fully aware of the risks and benefits of a procedure before agreeing to it. However, germline editing complicates this principle. But how do we obtain consent from someone who doesn't yet exist? This is a fundamental challenge with germline editing. The individuals who will be most affected by these genetic changes are not yet born and cannot voice their opinions or concerns. Germline editing poses a unique challenge, as the individuals who will be most affected by these alterations are not yet born and therefore cannot provide consent. This raises questions about the rights of future generations and our responsibilities towards them. Moreover, germline editing raises the specter of designer babies. The idea that parents could choose specific traits for their children, such as eye color, height, or even intelligence, is both fascinating and troubling. While the technology might initially be used to eliminate debilitating diseases, the potential for misuse is significant. The line between therapeutic and enhancement purposes could become blurred. It could eventually be used to select for desirable traits, such as intelligence, athleticism, or physical appearance. This could lead to a society where genetic enhancements are a privilege of the wealthy, further widening the gap between different social classes. This could lead to a new form of genetic discrimination and exacerbate existing social inequalities. Those who can afford genetic enhancements may have significant advantages over those who cannot, leading to a divided society. The ethical implications of germline editing are far-reaching and demand careful consideration. We must weigh the potential benefits against the risks and ethical concerns. We must engage in thoughtful dialogue about the potential consequences of altering the human germline and establish clear ethical guidelines to prevent its misuse. This dialogue should include diverse perspectives to ensure that the voices of all stakeholders are heard and considered. Section 6. Designer babies, navigating the slippery slope. The concept of designer babies, once relegated to the realm of science fiction, is now within the realm of possibility thanks to gene editing. While the idea of eliminating genetic diseases is appealing, the prospect of using gene editing for enhancement purposes raises serious ethical concerns. The slippery slope argument suggests that allowing gene editing for therapeutic purposes will inevitably lead to its use for enhancement. Once we start manipulating genes to prevent disease, where do we draw the line? Will parents feel pressured to enhance their children's intelligence, athleticism, or physical appearance to give them a competitive edge in life? This raises concerns about genetic discrimination and the creation of a genetic underclass. Those who can afford genetic enhancements could have a significant advantage over those who cannot, exacerbating existing social and economic inequalities. The potential consequences of designer babies are profound and demand careful consideration. We must engage in open and informed dialogue about the ethical implications of genetic enhancement and establish clear boundaries to prevent the commodification of human life.